This recorded session is dedicated to our short overview on our module about gene structure and function. So last Monday we weren't able to meet, so I'm recording the lecture for the benefit of you two. So please go over or screen this recorded session as you proceed with completing this module. So the concept of the term gene or the term gene has been evolving. The definitions of this term as used in molecular and human genetics has been evolving over time. So we have three main views: the classical, the neoclassical, and the contemporary, the modern definition. Um, uh, since or starting early or late 1990s up to now. No? So the classical view of the term gene, which prevailed in the early 1900s until 1930s, it is considered as an indivisible unit that is responsible for the transmission of traits from the parent to the progeny, as well as the recombination of these traits, as there were observations in which, there were observations where the progeny possesses characteristics that are midway between the traits of the parents. Also, the classical view considers the gene as an important unit for an important unit that undergoes mutation over time and ultimately is responsible for the final product and its output. The final product referring to proteins or factors that relate to um, transcription and translation of the protein product. No? When there was discovery, or when eventually when it is discovered that there are phenomena such as gene insertions, gene deletions, as well as gene duplications, which all contribute to changes, physical, structural changes on the genome of an individual. There's a new conception that came out. No, there's a new definition that came out for the term gene. No, so this neoclassical view prevailed until the 1970s, and this definition covers or describes the gene as multimodular or modular, can be subdivided into constituent parts. No, and these constituent parts have specific functions or specific um, roles that ultimately will contribute to the final expression of the product coded by the gene. No? And during this time, the each gene is conceived as a single producer, a single unit that produces a single product. So one gene is transcripted to a single mRNA, and that single mRNA is translated as a single or as is translated to a single polypeptide chain or a single protein molecule. No? So from the 1950s to the 1970s, the neoclassical definition of of the concept or of the term gene has prevailed. No? Then starting from 1970s, as we begin to have more technologies that allowed us to explore the structure of a living system's DNA, we're able to have a modern conceptualization of the term gene, wherein the classical and the neoclassical definitions are now are not enough anymore to totally define it. No, so the modern definition already includes interactions of the gene with other genes or with other genes or um, allelic forms of that specific gene. No? And we came to know about transmission patterns. And we also identified from the um, advent of newer DNA technologies such as PCR and DNA sequencing, no, the Human Genome Project, if you would remember from our previous modules. So with these technologies, we came to understand more about the structure and function of different genes, and we came to understand that it's not just a simple, or it's not the simple, the basic concepts that we used to subscribe to during uh, the first three-fourths of the 1900s are not enough anymore to really cover the essence or the ethos of the term gene, no? So um, the we know to date, no, or on the interaction, the final the in terms of the expression of genes, no, we um, we are introduced to the phenomenon that genes interact with other genes, or they may interact, and this interaction actually affects how they are finally transcribed and translated, no? So the interaction may occur between alleles of a single gene, and we call that allelic interaction, or the interaction could be between two or more genes that came to be, or that uh, later on was termed as non-allelic interactions. So these gene interactions, as we understand them, is actually a fundamental deviation from the one factor, one gene, one product, or one factor, one product idea of genes in in the classical and the neoclassical viewpoints. Okay. But there are different types of allelic and non-allelic interactions of genes in humans. So please take note of the ones that are in of the types listed in table number 5. Okay, and some examples for each. Okay. Now, we go into gene expression. Okay, so we will discuss transcription and translation as two key processes. So transcription is when RNA is synthesized from DNA. Okay, the process is controlled by structural units within a gene or an operon called promoters, enhancers, or other types of regulators. So review that there are different types of RNA that are produced, such as mRNA that is later on translated as a protein product, a transfer RNA, which is important for carrying specific amino acid residues that elongate the polypeptide chain, and ribosomal RNA, which is important, which is an important component of the protein translating machinery of ribosomes. No? So all of these three interact during translation. No? Now, for the case of transcription, there are four major steps that are involved. So there is initiation, elongation, termination, and post-transcriptional modifications or post-transcriptional processing. So initiation, review that this is when the DNA molecule unwinds, so it forms a bubble, a transcriptional bubble, where as an open complex, it allows the binding of a specific enzyme, the RNA polymerase. The RNA polymerase binds to the promoter region of that specific gene on the template strand, and when that event is cued to be successful, the next step begins, which is elongation. So elongation is when RNA polymerase moves along the template strand of the DNA and will synthesize an mRNA molecule. Okay. In bacteria and in non-eukaryotic, in prokaryotic cells, the RNA polymerase is actually a hollow enzyme. It is consisting of several subunits, including an important component called the sigma factor allowing the bacterial or the prokaryotic RNA polymerase to recognize the promoter. In eukaryotes, there are different types of RNA polymerases. So there's RNA pol 1, 2, and 3. No? And unlike bacteria or like in prokaryotes, eukaryotic transcription has an additional proofreading mechanism or proofreading step that ensures that the mRNA transcripted is faithful to the template DNA strand. Then the third step, which is termination, is when there is dissociation of the RNA polymerase with the template strand, and there's dissociation of the RNA polymerase from the open complex, allowing for freeing of the transcripted mRNA. So in prokaryotes, termination can be approached, or there are two approaches or two mechanisms in which it ends, no? Or in which termination is carried out. So there's what we call row-dependent termination and row-independent. So in row-dependent termination, the protein factor called Rho is responsible for disrupting the complex involving the template strand, the RNA polymerase, and the RNA molecule. In row-independent termination, there's no protein factor Rho, and what completes or terminates or ensures termination is a structural product or a structural no a structural unit. So a loop forms at the end of the RNA molecule, and that will cause the detachment of that RNA molecule from the transcriptional bubble. No? Termination in eukaryotes, on the other hand, it's a more complicated event which involves the addition of nucleotides, particularly adenine, at the 3' end of the transcript, and that's what we call polyadenylation. And post-transcriptional processing is when the RNA molecule is further processed into based on several or following different things, or to ensure that the final transcript is usable. So introns are removed because introns are the intervening components within the transcript. They're not really coding for anything, so they are removed. The exons are the expressing components, which are spliced together. So in between exons, there are introns. Once introns are removed, exons are spliced together to form a mature mRNA transcript, which will be responsible for coding a single protein. No? Then, following RNA synthesis, the process follows the normal base pairing rules. No? But the difference is the base thymine is not used, and it's replaced with the base uracil. Okay, so let's have some visuals for reinforcement of learning. Let's take a look at transcription. Okay, so I mentioned this already. Okay. Let's see. So the components, the template strand, is the strand from which the RNA is actually transcribed, and we also call it by the term antisense strand. Okay, so here, see, from the template strand, after transcription, you have an RNA molecule with complementary nucleotides to the template strand. Take note that there is what we call the process of there's a specific description for the process of transcription. Since only the template strand is used for transcription and the coding strand is not, no, both strands can actually be used as template. So you have to follow the direction at which RNA polymerase adds nucleotides to the nascent RNA transcript. No, so they can actually the process of transcription can actually be initiated on opposite directions. So here, so we add okay, so here you can see that the template strand, right, from 3' to 5'. So that's the order of the original what they call the original direction for the template. So it produces a 5' to 3' RNA molecule. The coding strand, on the other hand, since at this planar view, okay, if the if the what they call this, the RNA polymerase will read this gene or this part of the gene from this end, from the 3' end of the template strand to the 5' end, it will be able to transcribe it. But the coding strand, on the other hand, it starts with 5 primes, so the RNA polymerase will not be able to use it as a template. But if you reverse, if let's say for example that the RNA polymerase and the promoter is on this side, that I'm the one that I'm highlighting right now, you expect that the original coding strand will be the template strand since it's running from 3' to 5'. And the RNA polymerase will be able to provide transcriptional services to this coding strand, or should I say the template strand in this direction, following this direction. Okay? Alright, so that's what we call asymmetric transcription. Okay, so this one is discussing that, or helps you visualize what I have explained the last 40 seconds. Okay. Right. So now let's take a look at the RNA polymerase. So the prokaryotic RNA polymerase is a holoenzyme with multiple subunits. Subunits fall or are recruited as needed. They are removed when they are not needed. Total of 480 kilodaltons. Eukaryotic systems, on the other hand, have 3 types of RNA polymerases, each of which is a multiple subunit protein and are responsible for the transcription of the different types of RNAs. So here is the RNA polymerase of the bacteria E. coli, which is a holoenzyme. Take note of the subunits and their specific functions. Okay. Now for eukaryotes, take note of the following. So RNA polymerase 1 transcribes ribosomal RNA. Okay. 45S ribosomal RNA. RNA polymerase 2 responsible for hnRNA transcription. RNA polymerase 3 for ribosomal RNA, transfer RNA, and snRNA, small nucleolar RNA. Okay. So each transcriptable region in the operon, or each transcriptional region that is responsible for a specific product or products, is called an operon. And one operon includes several structural genes and regulatory sequences that are upstream to that structural portion, to that transcriptable region. The promoter is the DNA sequence where your RNA polymerase binds, and it's key in the control of transcription. So here, regulatory sequences are upstream to the structural components. So RNA pol attaches to these regulatory sequences. Okay. For prokaryotes, the prokaryotic promoter, there is what we call a consensus sequence, where which defines the promoter section. The -35 region, the Tata-GACA, and the -10 region, or the Pribno box, the Tata-ATA. Okay, so this is the promoter sequence, or the promoter region, and your structural gene starts at 1. Okay, at base or position 1. Here you can see some examples for a specific different types of genes, and the -35 and the -10 region. So you see consensus in these areas, no? That define the promoter sites. Okay, on the -35 region, the Tata-GACA is the recognition site and the initial binding site of RNA polymerase. And the minus 10 region, the Tata-Ata, is the region at which there is already a stable connection or complex between the DNA molecule and the RNA polymerase. Okay, now let's take a look at uh, the transcription process. So there are three phases minus post-transcriptional processing. So initiation, elongation, and termination. Okay. So here I mentioned this already. Okay. Alright. Let's take a look. Okay, so here, so yeah, here. So binding, your RNA pol binds to the promoter site, minus 35 to plus 1. Once a complex or once there's perfect binding, RNA pol establishes stability at minus 10 region, will now initiate opening of the DNA helix. Opening will now allow for reading of the antisense strand, okay, so that an RNA transcript colored green here will keep on elongating. Now, and then eventually it will be terminated. For bacteria, there are two approaches I mentioned already: the raw dependent and raw independent termination. Okay, for bacteria, take note that since it's a prokaryote, there's no compartmentalization of RNA transcription or translation. There is actually simultaneous transcription and translation in bacteria. So meaning single DNA molecule can be or yeah, single DNA molecule, um, several regions of it are actually involved in these two processes at the same time or simultaneously. So you can expect that that while transcription is happening, translation is also happening at the same time. So unlike in eukaryotes, transcription can be compartmentalized inside the nucleus, and translation happens outside or within the cytoplasm. But for bacteria, it happens in the same site, in the protoplasm of the bacterial cell. Then for terminations, we have raw dependent and raw independent for the case of bacteria. So the raw protein, which is a ATPase, yan, a hexamer, no? Which at the same time is a helicase. What it does is it once it is present, it recognizes the need for or it envelopes or it becomes a backbone or core of the growing transcript, and once it catalyzes the reaction of dephosphorylating an ATP molecule, then it's time that termination will be started or termination should be carried out. Raw independent, on the other hand, doesn't involve the raw factor, the raw protein. The termination signal is a stretch of 30 to 40 nucleotides on the RNA transcript, primarily made up of GC, followed by a very long series of U bases. The sequence specificity of the nascent, the growing RNA transcript, will then form particular stem loop structures that will terminate physically the transcription process. Okay, so here, the loop formations, and the hairpin loop formations will prevent transcription or will stop transcription. Here you can see. So once that the hairpin structure forms, it's now time for transcription to terminate. Okay? All right. Okay. So, here let's take a look at transcription in eukaryotes, since there are a number of things I want to discuss further. So for eukaryotes, initiation needs promoter and upstream regulatory regions. No? Cis-acting elements, which I'd like to introduce, are the specific sequences on the DNA template regulating transcription of one or multiple genes. No? Cis-acting elements uh, are as follows. So we have the GC box, the CAAT box, and the TATA box. So these six acting elements, similar with the promoter region, so -35, -10 for bacteria. So it's homologous to those areas in the prokaryotic system. Okay, and these cis acting elements are upstream to the structural gene component that is later on transcribed as mRNA. So here we have the Tata box, which has been identified as a consensus sequence in different types of organisms or eukaryotes, to be specific. No? So for eukaryotes, the RNA polymerase, unlike in prokaryotes, okay, in eukaryotes, the RNA polymerase does not directly bind to the promoter region, to the enhancer promoter regions. Okay, RNA polymerase associates with different types of transcriptional factors. So we have transcriptional factor 2A to 2H. So these trans-acting factors are proteins are recognized and bind directly or indirectly to cis-acting elements, and then and are considered as the ones responsible for regulating activity. Okay. Now, there are a series of events that factor these transcriptional factors, these trans factors. Won't really require you to review them since this is more generic, no? But just remember the fact that in eukaryotes, the initiation of transcription is more complicated, more multi step. Elongation is very similar to prokaryotic systems, and unlike bacteria, transcription and translation are compartmentalized. They do not take place simultaneously since they are separated by the nuclear membrane. For termination, the termination sequence for eukaryotes is long tail of A, polyadenylation, followed by multiple GT. A termination is closely related to post-transcriptional modification for eukaryotic organisms. Here. So from your growing RNA, that's later on processed further, the 5 prime cap and the 3 prime tail, the poly A tail. Okay. And let's go back to translation. Translation is the process wherein your mature mRNA molecule will be used as a template to assemble polypeptides, series of amino acids that will correspond to the primary sequence of a specific protein. And this involves 4 steps, namely initiation, wherein small subunit of the ribosome binds at the 5' end of the mRNA molecule and moves in 3' towards a 3' direction until it meets a stop codon. Until it meets a start codon, which is AUG. It then will form a complex with the large unit of the ribosome, the ribosome complex, and an initiation tRNA molecule. Elongation proceeds where subsequent codons on the mRNA molecule will determine specific amino acids. Okay? An enzyme called peptidyl transferase will link the amino acids together using peptide bonds or by synthesizing peptide bonds. The process will then continue until you have a chain of amino acids that moves along the mRNA molecule, and when when the ribosomal complex encounters a stop codon, a UAA, a UAG, or a UGA, translation comes to a halt. The ribosomal complex in eukaryotic organisms is much complicated. It's larger and more um, um, uh, more regulated compared to prokaryotic systems. In addition, the processes of transcription and translation in eukaryotic systems I've mentioned already are divided or compartmentalized, and eventually your nascent your dissociated mRNA will undergo post translation, or sorry, I mean your translated protein will undergo post translational processing depending on its nature. If it's a secretory protein, actually for both cases, secretory or a cytoplasmic protein, no, normally or we expect that the post-translational events will happen in the endoplasmic reticulum. No? Let's review translation. Okay, so let's review the here. Translation involves the following components. We have the mRNA, the ribosomes, and its corresponding or associated ribosomal RNA, transfer RNAs, and the codons. Okay, the codons, you'd remember we have a specific genetic code table. So we have the first mRNA base on this side, the second mRNA base on top, and then this one is a third mRNA base. So specific three-letter combinations of the different the different RNA bases correspond to a specific amino acid. Our start codon is AUG, methionine, and our stop codons, take note, we have UAA, UAG, and UGA. Let's take a look here. Alright, so here you can see from translation, so we read the following codons. UGG corresponds to TRP. So there's TRP here. Japan. UGG. GG. Okay. Then we have UUU corresponds to phenylalanine. UUU. Okay. Then GGC, amino acid GLY, G, GC, GLY. And we have UCA, which is corresponding to serine. UCA, corresponding to serine. Okay? Now, I want you to take note that there are codons that are considered degenerate, meaning they are different types, or they're different by one letter, one or two letters, but they correspond to the same amino acid. Like for the codons that are coding for GLY, so take note. There are 4 codons coding for it. Okay. Then we have the transfer RNA molecule. The anticodon region is the 3 RNA bases that matches the 3 bases of the codon on the mRNA molecule. So this one's highlighted in cyan are considered the basis of the anticodon. The ribosomes, which facilitate specific combination of tRNA anticodons with mRNA codons during protein synthesis. Okay, so the ribosome has 3 binding sites. We have the P, A, and E sites. So the P site is the peptidyl tRNA binding site. The A site is the amino acyl tRNA binding site, and the E site is the exit site. Okay, so we have on the small subunit of the ribosome is the mRNA binding the mRNA binding site. So, right, tRNAs that have been used up and the amino acids that they carry. Once it's attached to the nascent polypeptide, will be ejected from the complex. So our peptidyl tRNA binding site, this one, contains the nascent, the growing polypeptide. Our amino acyl tRNA binding site contains the new tRNA or the entering tRNA. that's not yet bound, or that the who's with the amino acid residue that's not yet um, bound to the growing polypeptide. So we'll just keep on producing the bonds or connecting the amino acid residues with the bonds, the peptide bonds. Okay, so I think that's all for translation. No, I won't discuss this. I mean, the remaining slides, I will just talk about the same thing about translation. Okay. Now, for gene regulation, so it's important as a phenomenon. It's important for the utility, the multiple utilities of, or the multiple products that are produced by a single group of gene, a single coding gene. No, so it will allow for modifications of the product as needed, um, production of different homologs of the same protein, no, and essentially the process will the process will dictate the rate at which proteins are produced, final products are produced, no? And these processes or mechanisms that are important for gene regulation, or that are important in carrying out gene regulation, uh, cover the following or include the following. When the mechanisms involve regulating the rate of transcription, like how fast transcription should be, how slow should it be, depending on feedback loops, which are usually dictated by concentrations of the product and the need for product present at the moment. No? Mechanisms, another one is mechanisms that regulate processing of RNA molecules, such as alternative splicing, so that you can produce more than one protein product from a single gene. Another is mechanisms that regulate stability of the mRNA molecules, something to do with RNA breakdown. So if the mRNA is not needed anymore because there's too much of the product it codes for, then it can be broken down so that the molecules can be recycled for other purposes. And ultimately, the last type of group of mechanisms regulating or uh, affecting gene regulation are mechanisms that regulate translational rates. Okay, so that ends our lecture on gene structure and function. Just a reminder, you have your last quiz on for the midterm, your last enabling assessment, your case study on gene expression and regulation. There are 5 questions, 10 points each. The due date for this is tomorrow. So upload a copy of your submission via the comment section of the offline assessment. So here, you can just comment here the link of your submission.